Hey guys, so today we are going to go beyond Mendelian genetics. So we're going to start looking at some of the different kinds of inheritance that actually happens. Um, Mendelian inheritance is actually really uncommon. Most traits are handled by multiple genes or are more complex than just like two alleles. You have this color or this color, and then one's dominant over the other. Most of them are more complicated than that. So we're going to start by quickly reviewing Mendelian, which is what we did last week. The Mendelian inheritance is one allele completely dominant to another one. So you've got a dominant, you've got a recessive, that's it. So the examples, a lot of what we looked at last week was Mendel's pea plants. So we looked at how purple flowers are dominant over white. If you have any copies of the purple gene, the flower is going to turn out purple. You got to have 2 copies of the white gene for the flower to turn out white. So that's what we did last week. But this is really not the case. So oftentimes traits are not a matter of dominant and recessive alleles. After Mendel's studies, scientists realized that there were many instances where Mendelian genetics could not explain inheritance patterns, and most of the time the patterns were more complex. So a good example, and we'll get into this later in the week, is blood typing. Blood typing is what's called a codominance. So we're going to get into things like that. But we're going to start with this idea of polygenic inheritance. Poly means many, and genic means genes. So it just means that multiple genes are governing a particular trait. One of the easiest ones to talk about is skin color. So down here on the right-hand side, there's actually about 3 genes that turn out to be skin color, and they work together to create your skin tone. So that's why there's so many different skin tones out in the world. In this chart, I feel like does a really good job of showing that. So you've got what they've classified as the A gene, the B gene, and the C gene, and different combinations of those alleles will give you slightly different skin tones. So you've got all the way down to the fully recessive, 2 little A's, 2 little B's, 2 little C's being the lightest skin tone you can end up with, and then the dominant genes being 2 dominants, 2 dominants, 2 dominants, giving you the darkest skin tone on the map. It also shows you down here at the bottom the ratio of the most common types of skin tones. So this, what I would call a caramel color in the middle, is the easiest to attain because there's the largest number of combinations that will give you that color. So if you look this right here in the middle, this complete heterozygote, so big A, big B, big C, gives you that kind of caramel color. But so does this one, which is also a heterozygote, and so does this one, and so does this one. So when we look out here on the sides, you can actually see the parents that are being crossed and all the different parent combinations that could be handed off. And the most common combination to come out of these crosses is this heterozygote, this full heterozygote cross. So it's complicated. That's why when we start talking about, we'll get questions about, well, my parents look like this, why don't I look like that? A lot of times it has to do with something that's more complicated. There's more genes going on. We're not going to do these polygenic crosses because they're a little crazy. And we'll get into what's called a dihybrid cross later, and there'll be a little bit of extra credit. But I just wanted you guys to see that most genes are actually a polygenic inheritance. So the next thing I want to show you guys is this idea of a bell curve. So it says bell curves can be created from data gathered using polygenic traits. If a population of human height is sampled and recorded as a normal distribution of bell curve graph will result. So this is called a bell curve. This is when we talk about normal. Normal is a statistical word. So if you are within, typically we say 2 standard deviations. So this middle here you can see is 0. If you're within the first 2 lines, we usually consider you to be normal, within the state of normal. Once you get outside of those standard deviations and you're out here in these little tails, then you're outside of the normal. That's really all that means. We're not going to spend a ton of time looking at those curves. So what we are going to do some practice with is this idea of multiple alleles. So it says there's more than one allele available for each gene. The easiest one to talk about is blood type, but actually not the examples I'm going to give you first because we're going to talk about blood type later this week. But there are 3 possible alleles you've probably heard of with blood type. There's A type, B type, and O type. They're all part of the same gene, So instead of being A or B, you can be A or B or O. So there's 3 options instead of 2. It's important to note, let me get my face out of the way here, that although there are multiple alleles for certain traits, an individual is still limited to 2 alleles per gene, just like anything else. So remember, you still only get 1 copy from your mom and 1 copy from your dad. So just because there's 3 options doesn't mean you have 3 genes. You only have 2. There's just more to choose from. It's like having more pairs of shoes. You know, you can only wear 2 shoes because you've only got 2 feet, even if you've got more shoes in the closet to choose from. So when we start looking at how to solve these problems, I'm going to get rid of my face so it's out of the way here. When we start looking at how to solve these problems, the first thing we usually need to do is create what I would call a dominance spectrum, because some alleles are still more dominant than other alleles. The most dominant allele is still typically represented with a capital letter. So we've got this in rabbits. We have different rabbit or chinchilla rabbits. These are rabbits, right? Yes, rabbits. Rabbit fur color. So you've got what's called wild type. You've got this chinchilla color. You've got this Himalayan, which is the white with the tips. And then you've got an albino that's all white. On the dominance spectrum, this capital C, which we have known now to be dominant, is called wild type and is the most dominant fur color. So it will override any of these other 3 colors. The chinchilla type will be represented as C with this CH exponent, basically. It's called a superscript. CCH is recessive only to the wild type. So it'll get covered up by the big C, but will override this Himalayan and will also override the albino. So it's in the middle of what we call the dominant spectrum. The next type is the Himalayan, which is dominant only to the albino. So it will get covered by chinchilla, it'll get covered by wild type, but this Himalayan is dominant to albino, so it will cover up albino. The last is albino, which is recessive to everything. So the only way to end up with albino fur, you can see right here, is this little c genotype, because it's recessive to everything else. You're like, okay, that's a lot. So let's look at an example. Where are my example problems? Okay. So let's say we're going to cross these 2 parents. We're going to cross this Himalayan because we see CHC means that they are Himalayan, but they're going to carry for the albino coloring. We're going to cross them with this chinchilla colored rabbit who has 1 chinchilla gene, but also has 1 Himalayan gene. They've got the CH instead of the CCH. So both of these guys would be heterozygotes because they don't have 2 of the exact same types of alleles. They've got a combination of alleles. But remember, you go with that dominant spectrum to find out what they're going to look like. So the CH is dominant over the little c, so you end up with the Himalayan color. The CCH is dominant over the CH, so you end up with the chinchilla color. So when you go to cross them, you set your parents up just like you would. So you split the alleles over the boxes for each parent, and then you run your cross. Again, put your dominant trait in the front. So whichever of the 2 traits go into that box are dominant, put it in the front. So when we go to look to see what kind of offspring, you always look at your dominance spectrum here. CCH is going to be dominant over that CH. So this offspring would be chinchilla colored. This offspring, CCH, will be dominant over that little c. So this one will also be chinchilla colored. This one is your only homozygous. It ended up CHCH, which is going to be Himalayan. And then this one is CH and then just plain little c. So it's Himalayan and carries for albino. It's really not all that different from the Mendelian crosses. You just have to think about this spectrum of dominance. What's the most dominant versus the least dominant? So when we look at phenotypes, half of these offspring, these top 2, would end up chinchilla colored, and the bottom 2 would end up Himalayan colored. So that's how we run multiple allele crosses.